Hi everyone, I'm Charlotte Kilpatrick, reporting for VaccineNation from the World Vaccine Congress in Barcelona. Today I'm delighted that we're joined by Dr. Ofer Levy of Boston Children's Hospital. Thank you for your time. Would you kindly introduce yourself and your current role?
Sure, and thank you, Charlotte, for hosting me here. My name is Ofer Levy. As you mentioned, I'm director of the Precision Vaccines Program at Boston Children's Hospital. I'm a professor of pediatrics at Harvard Medical School, and I'm also an associate member of the Broad Institute of MIT and Harvard. I also have a status as a special government employee of the United States, serving on the FDA, Food and Drug Administration Vaccines and Related Biologic Products Advisory Committee. Of course, my comments today are my own personal opinions, not official FDA pronouncements.
Fantastic,
Yeah.
thank you. So, your first session explored mucosal immunity. Can you tell us about some of the key challenges in this area?
Yes, well, it's important to keep in mind that the human body's immune system is multi-layered. And a key layer is the mucosal layer, our mucosal surfaces, including our oropharynx and our respiratory tract, etc., nasopharynx, etc. And that's often where pathogens, such as a viral respiratory infection, first infects our body. So the mucosal immune system is very important. The question is, can we develop vaccines that reliably induce mucosal immunity? There are some, like the Flumist intranasal you may have heard of, but rather few vaccines engage that pathway, that route of immunization. And the idea is to induce, for example, immunoglobulin A. It's a type of antibody that's on mucosal surfaces. And the importance is it might be able to not just prevent disease, but prevent infection and transmission to other people. So that could be a big win, but the preclinical models for mucosal immunity are challenging. And so, at the session, a great progress was presented, a lot of innovative approaches towards mucosal vaccines, but the jury is out in terms of how many of these will make an impact. It's a very important effort, and we talked about the challenges and the importance of the work ahead. Yeah.
Fantastic.
Yeah.
You presented in immune profiling covering human in vitro modeling for age specific adjuvanted vaccine discovery and development. Can you tell us about the significance of age specific vaccines and give us some context in broader precision vaccination?
Sure. So, human infections, and especially infections that lead to death, are not randomly distributed in the population. And one of the biggest demographic features that predict your risk of severe infection is age. So if you imagine a graph where the y-axis is the number of infectious deaths and the x-axis is the age of the humans who are dying of those infections, it's a U-shape. In other words, very early in life, in newborns and infants, there's a heavy burden of infection-induced mortality, and then this rises again as we age, as our immune system weakens. And so we want to keep an eye on those vulnerable populations and make sure we're developing vaccines that are tailored to work well in those populations, because those same distinct immune features that render those young and elders susceptible also make them more challenging to efficiently immunize. And so we've developed model systems where we can model immunity outside the body in a human-specific, species-specific manner, in an age-specific manner, working with blood-derived white blood cells and blood plasma, and then are able to test multiple adjuvants at different concentrations or combinations of adjuvants to identify those that might work best in a particular age group. And it does vary dramatically by age.
Fantastic. So you've mentioned the modelling already. Could you tell us a bit more about how this is going to contribute to these efforts?
Yes, absolutely. If those of us on this interview session were in a human clinical trial for a vaccine, we would each be assigned to a treatment group or a control group. But in the human in vitro system, if we draw some of your blood, for example, and test it in culture in a plate with different wells, we could test at the same time a vehicle control, in other words, like a placebo, different concentrations of adjuvant, different concentrations of antigen and combinations, and we can benchmark them to a vaccine vaccine that's licensed or approved and known to be safe and effective. And so now we can gather information about which adjuvant antigen combinations might work best in you, and gather biomarker information that might predict the risk of reactogenicity with a vaccine and the immunogenicity of the vaccine. And in this way, we might be able to accelerate and de-risk vaccine development. I mean, if you think about all the different types of infections we want to protect against, and then for each of those, there's a range of candidate antigens, and there are a range of candidate adjuvants, and there are a range of candidate vaccine platforms, and then you layer on that by age, sex, demographic features such as geography, where people live, all of those are variables. If you multiply all those variables out, there's not the time and money in the world to do large phase 2 and 3 clinical trials of every adjuvant antigen combination in every location on Earth. So if these in vitro systems can show some fidelity to generate information that's predictive, then that's a huge win to accelerate and de-risk vaccine development that's precise, it's tailored to the population.
Yeah, that's very exciting.
Yeah, it is.
So, I also wanted to ask about your fentanyl vaccine efforts. So, you're developing an adjuvanted fentanyl vaccine. Can you tell us why you're pursuing this and how it's going?
Yes, well, in terms of why we're pursuing it in the United States, for example, and the problem is more global than that, but let's just look at the U.S. for a moment. Currently, we have approximately 200 Americans a day dying of drug overdose, mostly fentanyl, which, as you know, is a synthetic opioid that's very, very potent. Even small amounts, a few grains of fentanyl, is a lethal dose, suppresses respiration. And by the way, many of the teens, for example, and young adults who are dying of fentanyl overdose do not carry an opioid use disorder diagnosis or any prior history of opioid use. They may be at a party, somebody convinces them to swallow a pill that unbeknownst to anybody has some fentanyl in it, and the next thing you know, an hour or two later, they're dead. And it's a tragedy. And so in this context, the U.S. National Institute of Health, NIH, awarded to David Dowling, who's a faculty member in the Precision Vaccines Program, and I, as co-principal investigators, a large contract to develop an adjuvanted fentanyl vaccine. There have been efforts in this area before, but what they noticed was a blip of antibodies that quickly came back down, and so the adjuvant science may be particularly important in that effort to get higher titers of antibodies that neutralize fentanyl and greater durability. And the idea is that you would immunize people who are at higher than average risk of a fentanyl overdose. Certain demographic features are known to put you at higher risk: age, sex, males maybe more, etc. And you can identify populations of first responders, potentially U.S. military, because opioids can be aerosolized and weaponized. So there's a whole range of populations that might benefit from that. So funded by NIH, we've discovered adjuvants that might be optimal in opioid-using individuals. Opioids alter your immune system, so back to the precision approach. We modeled the immune system of opioid users outside the body. We worked with my wife, Dr. Sharon Levy, who's the chief of the first division of addiction medicine at a pediatric hospital in U.S. history. Last year, Boston Children's Hospital created a division of addiction medicine for children. It's a sad thing that we need that, but we very much do. And so in her clinic, we recruited individuals and obtained blood samples and then tested these outside the body and compared to age-matched controls who were not exposed to opioids and saw a difference in immune response to different adjuvants. So we selected the adjuvant that might be optimal in that population. And the antigen is fentanyl covalently linked to CRIM protein, that's a large bacterial protein that's also in the Prevnar vaccine. It's a hapten approach because the small molecule fentanyl is not very immunogenic, so you solve that by covalently linking it to a protein. So we have the antigen, a subunit antigen, and then an adjuvant that's tailored for that population, and that's proceeding in preclinical development, formulation development with our partners at Inimune, Dr. Jay Evans. And that's been a very successful project moving forward. And then in the midst of all of that, I also wear a hat as a co-founder of a company named OVAX, OVAX Inc., that has recently come out of stealth mode, covered at Bloomberg Media, and our CEO, Colin Gage, was just on Fox News last week. And that company is going to focus on the development of opioid vaccines, including a fentanyl vaccine, to prevent overdose deaths. That's really the goal of the vaccine. If somebody who was immunized were exposed to fentanyl, the antibodies in the blood would bind the fentanyl, preventing it from crossing the blood-brain barrier, where it suppresses respiration and kills you. And that would be an important tool to reduce the death rate due to opioids. And we've also been doing studies with the Division of Addiction Medicine that suggest that whereas the U.S. has the higher burden right now of opioid overdose deaths, there are patterns of use in Europe that suggest that it's on the cusp of also having an emergency with this. In the U.S., the opioid crisis has been labeled a public health emergency. The Department of Homeland Defense declares it a greater threat to Americans currently than terrorism. So this is a real emergency at all levels in U.S. society. And so OVAX hopes to bring a solution for that, and we hope to be in clinical trials within a year.
Fantastic. Well, it's certainly a gargantuan task, but we're very excited to follow that progress. So can you tell us about recent changes from the FDA with regards to human in vitro modeling?
Thank you for that, Charlotte. Up until very recently in the United States, we were operating under FDA rules that went back to the 1930s that stipulated that drugs, vaccines would go into animal models before they go into human, which certainly made sense, and a lot of work in mice, as you know. It's been, and our program makes use of mice too. They're very handy, they're very practical. That said, the innate immune system of mice is hyperdivergent from a human immune system, so mice don't always predict responses to adjuvants and adjuvanted vaccines accurately for humans. And in this context, there's been growing interest in human in vitro modeling. Can you model vaccine responses outside the body? And with the support of the Bill and Melinda Gates Foundation and NIH, the Precision Vaccines Program has been working now for over 10 years for modeling age-specific immunity outside the body. And we were very excited to learn that on the 29th of December, 2022, U.S. President Joe Biden signed the FDA Modernization Act 2.0, which now provides for the first time for the use of human in vitro modeling, systems biology, biomarker discovery, and artificial intelligence and machine learning as part of a drug and vaccine filing. And so we are working with our partners. We collaborate with industry. Multiple companies now have turned to us to help them accelerate and de-risk their adjuvant and vaccine development. And by the way, these model systems can also model mRNA vaccine responses. We have a manuscript now in press in the journal iScience about that, and maybe we can present those findings in Washington, D.C. in the year ahead. So I think the world is changing at FDA. New opportunities are opening up, and access to high-quality population-specific and age-specific human in vitro modeling will become an important part of the portfolio for vaccine developers.
That brings me nicely to my last question about your key impressions or lessons from your time at the Congress this year.
Well, this is a very, very important conference for us as a precision vaccines program, bringing precision medicine to vaccinology. World Vaccine Congress, we view as one of the preeminent vaccinology meetings in the world, the strong academic component, but also government agencies and industry, and as you know, a very strong industry component. And that's very important. We need to bring together those different perspectives of discovery, development, and clinical trials. And World Vaccine Congress does that to a high level of excellence, a high level of diversity, and we always look forward to your conferences every year. As you know, Precision Vaccines Program has a strategic partnership with Terrapin, and we are also developing our International Precision Vaccines Conference in Vermont, Burlington, Vermont, on the 8th and 9th of October, 2025, and you're all invited to that as well. We've been in touch with Wing about that as well. So it's been a great synergistic partnership. Our faculty present in the Washington, D.C. conference, for example. David Dowling, Joanne Arce, Simon Van Haren, other Precision Vaccines Program faculty have been presenting, and it's been very enriching and led to great collaborations.
Fantastic. Well, we're very glad that it was productive and look forward to the next one. Thank you for your time.
Thank you, Charlotte. Always a pleasure to be here.